Hi friends, today we will discuss about microfilaments in detail. These microfilaments or actin filaments, they are the major cytoskeletal proteins of most of the eukaryotic cell. Okay, and these actin molecules they polymerize to form actin filaments. Okay, these microfilaments they are the thinnest filaments and they are found in the cytoplasm of all eukaryotic cells. They are the linear polymers of actin subunits. So several actin subunits polymerize to form a linear filamentous structure. Now these microfilaments they shows assembly in two general types of structure. Okay, the assembly and disassembly of actin filaments, their cross links either shows in bundle or in network. So when they form bundles, these filaments when they are closely packed in parallel arrays, they shows the structure bundles. Okay, and when the filament crisscrosses often at right angles and when they are loosely packed they form network. Okay, so see here these microfilaments. Here we will discuss about some key points and this slide we will be going to do again when we will compare the three filaments, compare the three cytoskeletons. So these microfilaments or actin filaments, there are two intertwined strands of actins. So several actin subunits polymerize to form actin filaments. So how two intertwined strands we will discuss. They are around 7 nanometer in diameter and the subunit of microfilament is actin. What is their role? They help in maintenance of cell shape. They can change the cell shape, helps in muscle contraction, cytoplasmic streaming, cell motility and cell division. So you can see here that two strands are intertwined to form a helical structure, forming actin filament. So look in this picture, these actin filaments which are also known as microfilaments, they are the helical structures. These helical structure, each subunit, each circle which you can see, they are actin. So several actins are polymerizing to form this filament. And these filaments, they are very flexible and having a diameter of around 7 nanometer. Okay, they can be arranged in linear bundle or two-dimensional network or three-dimensional gels. Okay, they are present in abundance in the cortex which is just beneath the plasma membrane. Okay, they are present throughout the cell but they are concentrated beneath the plasma membrane. So what we discussed that these actin filaments they are the major cytoskeletons and they polymerize, these actin subunits polymerize to form actin filaments. They are thin around 7 nanometer in diameter, present in abundance beneath the plasma membrane. They form the network and they provide mechanical support and movement of the cell surface. Okay, next we come to assembly and disassembly of actin filaments. Now these actin filaments, the single subunit is G-actin. Okay, the single subunit which you can see they are G-actin which is around 43 kDa and has ATPase power. Clear? Now Now these actin monomers, they polymerize to form what? They polymerize to form filament. So the simplest one is the G actins, which are the actin monomers. They are polymerizing to form F actin, and two F actin arranged in a helical manner and is forming actin filament. Clear? So each actin monomer, so G actin is what? G actin is actin monomer. Okay, and this G actin, it has tight binding sites which helps in head to tail interaction with two other actin monomers. I'll show you the picture. So actin monomers are polymerizing to form F actin, which is a polymer. Okay, now this F actin, so I draw here. These are the G actin. This G actin, it's forming dimer, then it is forming trimer. Okay, ultimately they are forming polymer. Okay, which is F actin, a filament. Two F actins, they are arranged in a helical manner to form actin filament. So as I said, here it's written as well that two parallel F actins, they twist around each other in a right-handed helical manner to form actin filament. And these actin filament is a polar structure. It has a slow growing end and a fast growing end. The slow growing end is the negative end, and the fast growing end, also known as barbed end, has a positive end. Okay, and this rate of polymerization and depolymerization, also known as dissociation, it is equal. That is why the length of the filament does not change, it remains same. Okay, now these actin monomers, they bind with ATP. When they are in ATP bound form, they add itself to the filament's plus end, and when they are in ADP bound form, they leave from the minus end. So individual monomers, which are the G actins, they move down the length of the actin filament. Okay, either adding to the plus end or leaving to the minus end. Now this movement of individual monomers down the actin filament, this process is known as treadmilling, and this treadmilling is maintaining the dynamic equilibrium of the cell. It is maintaining the dynamic equilibrium between the monomeric and polymeric form of actin. Now, this actin polymerization, it is a reversible process. Okay, there are some drugs which affect the polymerization of actin. Number one is cytochalasin D. What it does, it binds to the positive end of the filament and block their elongation so that no monomers can add itself to the plus end. Another one is phalloidin. This phalloidin binds to the actin filament and it stabilizes them against depolymerization. So depolymerization cannot happen when phalloidin binds to the actin filament. So see this structure. What you can see here, that this actin monomer, this is an actin monomer. It has a nucleotide, maybe ATP or ADP, in a deep cleft in the center of the molecule. Okay, in a deep cleft, it binds with either ATP or ADP, and this is actin monomer, which is G-actin. Now this actin monomers, they are arranged in a filament, okay, consisting of two protofilaments. So here they are two protofilaments. Okay, and these two protofilaments, they are attached to each other by lateral contacts, which helps them in winding around each other in a right-handed helical fashion. Okay, so all the subunits here is having the same orientation. And the third one is electron micrograph. So why I have placed this picture to show that these two protofilaments, which are F-actin, they are arranged, they are twisted around each other in a right-handed helical fashion. Okay, next is, now this is see how the actin polymers are added. So here in the first picture, what you can see, that the polymerization which is occurring here, the polymerization of pure actin subunits into filaments, they occur after lag phase. So in the lag phase, they are forming dimers and trimers and oligomers. And after the lag phase, they are polymerizing. In the second picture, what we see, that the polymerization is rapid. Why? Because there are preformed fragments. There are preformed fragments of actin filaments. Okay, already the preformed fragments of actin filaments are there, and this preformed actin filaments is acting as a nuclei, that means a seed. Okay, so you can see here in the second phase that the polymerization is rapid because preformed fragment of actin filament is already there, which acts as a seed. Clear? And there is a critical concentration after which there is no addition of the nuclei. That means after which the number of added monomers is equal to the number of dissociated monomers. Okay. Now these actin polymers, whether it's a microtubule or a microfilament, they shows polymerization and depolymerization. So polymerization is by the addition of monomers and depolymerization is by the removal of the monomers. And the rate of addition and the rate of dissociation is represented by K on, which is the rate of addition, and K off, which is the rate of dissociation. This is just a general idea. Now, the next step is nucleation. So, what do you mean by nucleation? Is that this helical polymer, which is formed, this helical polymer, it is stabilized by several interaction between the monomers. Okay, so in case where the two monomers are associated with each other, it is relatively weak. Okay, the association between the two monomers is relatively weak. But when third actin monomer is added, it is comparatively stronger. But when other monomers are associated with it, the association becomes more stable. Okay, So I said when only two monomers are associated, then the interaction between these two monomers is very weak, and when one more monomer is associated, when they form a trimer, the interaction is relatively strong. I write here relatively strong. And when several monomers are associated, then the interaction is more stable. Okay, so when this trimers are formed, they act as a nucleus. So when these trimers are formed, they act as a nucleus for polymerization. Okay, and the assembly of this nucleus is relatively slow process. And this slow process explains that this is happening in the lag phase. Okay, so I write here the assembly of trimer. It is a slow process which explains that the lag phase is seen during polymerization. Now, how this lag phase is reduced, we already discussed in last slide. The lag phase can be reduced when there is preformed fragment or preformed nuclei. Okay, when there is preformed nuclei, then this lag phase can be reduced or abolished, and a fragment can be formed in a faster period, right? Next is the critical concentration. So what is the critical concentration? Critical concentration is when the number of monomers that are being added to the polymer per second is proportional to the number of free subunit. That means K on, that means the number of monomers that is added is equal to the number of monomers that are dissociated. The ones that are dissociated will remain free. So the number which is added is equal to the number of free subunits. Free subunits means the ones which are dissociated. Okay, here the rate is constant. So at this concentration, the rate of subunit addition equals to the rate of subunit loss. Okay, this is known as critical concentration. So the course of polymerization is, it is divided into three phases: lag phase, growth phase and equilibrium phase. In the lag phase we already discussed. In the lag phase what we see, it is the phase where nucleation occurs. Okay, and the growth phase, it is the elongation of the filament. And in the equilibrium phase of the steady state, where the K on is equals to K off, there is the balance between the association or the polymerization and depolymerization. Clear? So we will be continuing with the microfilament in the next lecture. Thank you so much for your patience and cooperation. Please like the video and subscribe the channel for more videos. Thank you.